Okay, so let's go to the tutorial. I mean, if any other person has questions as we are going along with it, then let us know. If you are stuck somewhere, you can talk. Okay, so most of the files have been, most of the softwares have been downloaded for you. So first thing first, make your raw data folder.
Okay.
Press Ctrl L.
Ctrl L.
Yeah. Yeah. So Ctrl L is for clear. So it's going to, I don't want to be like the log is too much. So
Okay.
CD into raw data. Now go and copy your WGS commands from that. Just copy all of them.
Okay.
Yeah. Copy. And then go and paste it on the terminal. So it will start working immediately. Immediately there's an enter, like next line. It will work on the first one, go to the next one. That's how scripts work too. So when you copy everything and just paste, it will just start working that way.
Okay.
Does anybody have a question? Sorry, I'm going to use this session to continue to speak to everybody. So if you have similar issues, you can just talk. Or if you have a comment on one of the steps. Yeah.
Okay, thank you.
Yeah.
I think, yeah, the way you start, it will be easier then, because I was stuck at the duplicate, the remove duplicates side.
I would also advise that you start from scratch.
Okay, maybe
Sorry.
I'll be following you now.
Yeah, you can also just delete your files already so that we can have space, because we are not going to create extra space for you. I mean, if we are going to create extra space, it will be for the project. This thing is not supposed to take more than 50 gigabytes for each person. I know it's not up to 50. It's more than, it should not be more than 10 gigabytes for each person. But some people are already taking way more than that. You can actually check and see those that are taking it, and I will paste it on the server.
I hope it will not log me off.
If it logs you off, you log back in. But don't worry, it's working. I mean, it's not is it a problem if you are logged off?
No.
Yeah, you can come back in.
When I log in at first, I was using my screen. After everything it will still remain on the screen.
Yeah, did you get out of the screen?
No, I have not.
Yeah, nothing is changing. You see that you just have to copy and paste it. It will be fixing it by itself. You see, this thing is easier than you are taking it. It could be way easier. You just have to be, how will I call it? You have to be more flexible with it. It's not so, and if you are stuck, always ask questions. But apparently you went ahead not asking questions, enough questions, I would say.
Yes.
You didn't ask enough questions.
I'm not always good at asking questions.
I think that's the most important thing you should learn. The biggest platforms for programmers is Stack Overflow. And what is Stack Overflow made of? It's made up of questions. It's made up of questions. So asking questions, knowing how to ask the right questions also matters sometimes, but asking questions generally is a very good skill.
Okay, sir.
There is also BioStars, and it's made up of.
Please, Sorry, how can I delete the whole folder of raw data without deleting each at once?
Without deleting?
Without going, maybe deleting the directory under the raw
You want
data
to delete everything under the raw data?
Yes.
Yeah, so you should delete it. Press enter.
Should be,
yeah.
I should use the remove directory. I've forgotten the code, like
RM.
What's it?
RM.
DRI.
No, RM. No, not RMDI. RM flag R. Then your raw data. It will delete everything.
Okay.
Do you know what you are doing right now,
Thank
you. Huh?
Do you know what you are doing right now?
Yes, I'm downloading the files.
Yeah. And the reason, so I just said you should copy the commands and paste it. So when you copy commands and there is next line, that is, there is like enter already in what you copied, it's going to start working immediately.
Okay. So there is enter already.
Yeah, for the last one, there was no next line.
Okay.
That's why you had to press enter by yourself to go to the next line and it starts working.
Okay.
So it's very good that you're able to reproduce a basic genomics pipeline, at least. At the minimum, you should be able to. If you cannot, it's difficult to say you are able to analyze genomic data sets. So we might take up to 2 hours on this call. If you are not ready for that, I would advise you go and sleep.
So 2 hours will be enough to do the whole thing.
Yeah.
So why are we spending 2 weeks? I've been on this.
I don't know. Because you are not asking questions. People spent 1 week or less on it. Okay. I mean, for starters, yeah. But you should not spend more than 1 week. The most important issue you are probably facing is that you are not specifying the right directories. If you are specifying the right files and directories, it will be really easy for you. So go back to the data set again. Let's see the link. So one thing you should notice is that you have 2 major samples. You have this sample and you have this sample, this T and the N. So for the T guys, they are in duplicate. They are this forward strand and the reverse strand. This one also has forward and reverse. Apparently, hopefully that's what it means. Yeah, I think so. So you have forward and reverse for these 2. Now, that means in all, you have just 2 samples, right?
Yes.
So you have just 2 samples. Now with your 2 samples, what you can, so you have to look at these 2 samples as one. I mean, these 2 pairs from the same sample as one. So I would mention this when you go forward, okay? But I just want you to know that you have 2 organisms or 2 sample or 2 cell lines, and you have forward and reverse data from the 2 of them.
Yeah. Okay.
I mean, this is the normal. Sorry, that was
sorry.
The N is the normal. I mean, you don't need to. The N is the normal, and the T is the probably 2 more.
2 more.
Yeah.
Please pardon me. Let me do this thing. It's making sound. I have to.
It's your computer. You can do anything you want.
Okay,
yes.
Now.
Yeah, you have all your data set now. Type LS first. Don't just be happy. LS. Let's first of all check even if it's downloaded. Then now press ls flag LH. It's a very important step. It's like just simple quality control. If you download a movie, what will you do? Or before you download the movie, you first of all check the file size, right?
Yes.
Yeah, so it's always good to check the file size so that you know I downloaded the complete movie or not. Now, go to your tutorial again. What's the next thing?
I should download the reference sequence.
Download the reference sequence.
So I'll copy it.
Yeah. I would say make a folder. Hold on. Make a folder first. Just press Ctrl C. Make a folder for ref. Yeah.
Is that okay?
Yeah. Then enter ref, apparently. Then paste your command there. Basically, what you are doing is downloading for now.
Okay.
Yeah. So with screen, you're actually using the internet on our PC here. You are not exactly using your own internet to download these things. You are just using your internet to look at a screen somewhere else.
Okay. So I'm working directly from your own computer.
Yeah, not from my computer, from the server.
Okay.
That's why it's somehow fast. But if you're using your own local computer, I don't think it will be this fast. And that's why that Google server, cloud server is also, and Google Cloud Share is also very fast.
Okay.
Because you are using Google's internet. And you can imagine the speed of Google's internet. So gunzip, I think that's the next step. Then HG19, just type H and press tab to fix everything for you.
Enter.
Yeah. That's the next thing. You don't need to wait for me. Yeah, it's done. So press Ctrl L. Let's shift all of this up. Yeah. So let's go to the tutorial. The next thing on the tutorial, so we've unzipped, so you don't need to, is to do quality checks. So for me, I would say don't do the fast QC. Okay. Because
okay.
either you like it or not, you will still come back to trim.
Okay.
Right? Yeah. So why do the fast QC twice when you know you will trim?
And still
by the ways, do you have SCP already? I mean, WinSCP to copy files from your terminal.
Yes, I do.
Can you copy files right now?
Yes.
Okay, that's good. So that when we get there, you will not be stuck. Okay. So let's try to remove low quality reads and just go to, we already installed it.
Okay.
We already installed it. So create, so I gave you a link. Do you have the link to download from Trimomatic directly?
Trimomatic direct. It was on Google.
Then you can look for it. Let's go to Google.
On Zoom. It was
Let's go to Google.
Zoom.
Let's go to Google on your computer. You will do everything. You will teach us today, don't worry.
Because it's not.
Yeah, Trimomatic. Just click on, Trimomatic. Just click on it already. You don't need to type it in full. That's why Google is there for you. Scroll. Yeah, Userdel Lab.
Okay.
The Userdel, I don't know.
This one is PDF.
Yeah, go back. This is not the website.
I think it should be this one.
Yeah. Then copy the binary source, either 0.36 or 0.69, anyway. The
this
binary, yeah.
or this?
Anyone, but the binary is better. Yeah. Get out of your ref.
Okay.
Then make, you don't need to make any directory. Download the data.
Okay.
Download. So you have to use WGS.
Oh. Sorry.
Yeah, it's your time.
Okay, it's done. I should go unzip. Unzip, right?
Yeah. You have to unzip. Press tab, yeah. You should be fast because we are trying to reduce time here. There are a lot of people that are waiting on you. Okay, so you have your Trimomatic files now. So my advice is that you copy, is it TrueSeq? So look at the tutorial. Let's look at the tutorial. So you compare what you have with what the tutorial, where the tutorial is going, so you can have a smooth save. Go to some, yeah. So they used TrueSeq PE, right? Can you see? Yeah. So they use TrueSeq PE, TrueSeq 3 PE.fe. So I will tell you, my advice is that you should copy TrueSeq PE, go to your directories, go to your terminal rather. Now, so you have this file in adapters. I'm coming, let me try to help you locate it. Yeah, this is it, right? If I'm right. Am I right?
Yeah, yes.
So what I want to do, go back to your command here. So what I want to do now is that, go back to your tutorial. Sorry. Tutorial, yeah. Your network is breaking. But yeah, so when you are here, what I want to do now is that I want to bring this guy to a place where our bash script will be, you'll be able to locate it immediately. Do you get my point?
Okay.
Yeah.
Yes.
So go back. Now say, so you have to copy TrueSeq PE from inside the Trimomatic folder to your present working directory. So do you know how to do that, or you need my guide?
Okay. CP.
Yeah. You have to copy Trimomatic. So I mean, you have to copy the file from Trimomatic. Yeah. Tab. Just use tab to make things faster. Yeah. Adapters.
Yeah.
Because it's located inside adapters. No. What is this? Then the TrueSeq file. So there are so many TrueSeq, so you have to specify the one you want.
Okay.
You still have two TrueSeq here. Yeah. So you cannot use autocomplete now. Then to your current working directory, use $,
Okay.
capital letter PWD. Yeah. Enter.
Enter.
Yeah. So type ls now. Now type ls flag lh.
So I think this is it.
Type the file size. Let's see if it worked. If we downloaded the true thing. You see, it's 93 bytes. Actually, it's not so. It's a very small file, so it's correct.
Okay, I'm afraid. I was afraid.
Okay, I mean, you can print it. Press type cat TrueSeq. Let's see what's there. So you probably also have to work on the accuracy of your commands because you're typing CSAT. You see, so something is inside. So
Something is inside.
yeah, like something is inside.
Okay.
Forward and the result. So clear it, Control L. Sorry, is that? Okay, so create your first script. That's the trimmer.sh. I mean, whatever you want to.
Okay.
Whatever you want to use, young man. Yeah, so let's go to our tutorial.
Excuse me.
Yeah.
Please, I have a question.
Yeah.
Because I've been following the tutorial step by step.
It's good.
And like towards that place where he said we should copy the trigonometric, I got lost there because it was saying no such file or directory. So
Yeah,
I don't know if I'm supposed to create. I don't know if I'm supposed to create an adapter directory.
Wait, did you download from where we downloaded?
Excuse me, sir.
Did you download?
What do you say? Yes, I downloaded the trigonometric.
Okay. Temitope, can you go over it again for her?
Okay.
Sorry, it's dragging you back though.
Sorry to bother you guys.
No, I'm learning too. Thank you.
He's happy to do it.
Okay. So,
like, I'm at that part where you just unzip that trigonometric. So I've done that one. Then when I was trying to copy trigonometric, that was where I had the issue.
So, sir, what did you say I should do?
You have also forgotten what I told you to do.
No, like the example.
She wants to copy trigonometric. She wants to copy the adapter file from inside trigonometric. So try to do. Yeah, you can exactly. I mean, you can just scroll up and see the old commands you have used.
Okay, that would be better.
You can also ask her if she has unzipped the trigonometric folder.
Yes, I've unzipped the folder.
Have you done this?
Which part?
So the last command there.
Press Ctrl L so that we can see.
Yes, I've.
Okay.
Yes, I've downloaded it.
Okay, then did you unzip?
Yes, I unzipped.
Okay, after that, did you follow this?
Yeah, this is the place I had the issue.
So what do you type in your case?
So I'm asking if I'm supposed to like create a directory before that time because it was saying that no such file or directory.
Which file did you tell it to copy?
The trigonometric.
Can you send your command here? Yeah, let her send her command here.
Okay, I'm sending.
Then she's probably specifying a wrong folder, a wrong file name. Yeah, Temitope, yeah, we are listening to you.
Okay. I think it should be the wrong file name. Maybe you should check the trigonometric spelling or the adapter or the TrueSeq. It's either of the three because we are downloading under our directory.
Can you ls? Sorry, please can you ls Temitope? Let me see if there is a file that I'm missing out.
We are using raw data, not Temitope.
Okay, ls raw data, please.
Okay.
Okay,
so this is
let me try to ls. Okay, I see the problem. I'm missing that TrueSec3 file.
Yeah.
Okay, that's the file we are working with. That's the file we copied into our raw data.
Okay.
Into our raw data directory.
So you have to copy TrueSec3 from inside the trigonometric and paste it outside. Do you understand what we are saying?
Okay. Let me try the command again.
Or paste the command you are trying on the chat, and then we just debug.
Start.
Paste the command you are trying, paste it on the chat, and then we can see it together.
Okay, let me do that.
Okay, Temitope, maybe you should go ahead. Let's continue.
Okay.
So let's go to the tutorial. So yeah, so if you look at the tutorial again, you have several things.
Yes,
And
sir.
Amila, we are still with you. Once you are set, just let us know.
But I've not created the.
Yeah, that's where we are going. One, you have to create trimmed reads. Hold on. I want to explain the command for you. Don't rush. You want to create trimmed reads. Now, there has to be a file called cat.list.txt, which you necessarily don't have to. Yeah.
Okay.
Did you just correct the code, or this was what we were trying before?
This was what I was trying before.
And you downloaded Trigonomatic itself? So your file is
Trigonomatic, exactly like that. I just copied it.
Temitope, you have to show her the command you used again.
Okay.
So she did not specify the right through sec3. Look at it very well and compare it with you. I'm not going to tell you the differences. I'm sorry.
Are you seeing it?
I've seen it.
Did you see the difference now? Look at it very well. Temitope, can you see the difference?
Okay, let me check.
Yes.
Yeah,
there's no P.
Right? Yeah.
There's no P.
Okay, I've seen the difference.
Yeah.
It's the capital letter E.
P. Bag P.
I think there is also a way to increase the size of this your screen. I'm not sure.
Me?
Yeah, your fonts are tiny. I mean, it's clear to me, but maybe for others it's not so clear.
Okay. Oh. I'm trying to see how I
Don't
can.
worry. It's before you log in that you have to do it, actually.
Oh.
So don't bother yourself about it. Daniela, you can look at it again and let me know.
Yes, I've seen it. Thank you. It worked.
Yeah, fine. Okay. Thanks to our teacher as well.
Oh.
Control C, or press Control C. I want to
Control C.
Yeah. Are you in the terminal? No.
Yes.
Yeah, you just came back. Yeah, you see, it went to the next line. Control C means cancel. So it's going to go to the next line.
Okay.
Okay, let's go back to our tutorial. So, if you make a directory, I mean, this we are going to make this. Hold on, don't go, calm down. You are going to make a directory. So we are going to create a script that will make a directory 1.
Okay.
Now we are not going to use this cat list.txt because this is where everybody started making mistakes. The first set of mistakes we saw, they were here. Now, what I want you to do now is to scroll up. No, scroll down then. I mean, this is supposed to be scroll down, but go up. Continue going until I tell you to stop. Yeah, here. So I want you to understand what is called patterns. Bash can understand patterns very well. So there's something here that is similar here and here. You see, up to this point, they are similar. In this case as well, up to this point, they are similar. So what you have to do is to just specify these guys, these things that are similar, and use it as your dollar, whatever. Oh, I'm unable to write dollar very well. And then you have them as maybe sample. So it is this pattern combined with this underscore, whatever you have, that the file will be looking for, that your command will be looking for. Okay, do you understand what I'm saying now?
Yes.
So I want you to copy these two, this one and this C version. Copy it.
Okay. Oh, should I
Your terminal.
Should I do it one? Oh, it can't.
End first. Go to your terminal. Create
I've created a script already.
And where is it?
Before I logged out.
We've deleted everything.
Oh.
I keep forgetting we've deleted everything. Sorry, we deleted everything. Did you see? Can you see now?
No, not the other one. Like before she asked that question, I thought I created a script.
When you type your ls here, did you see trim here?
No.
Yeah, so why are you looking for it? It's not there. Enter. So now paste. Hold on.
I should.
Yeah, so go and copy the T version as well.
Okay.
Paste, yeah. So you are supposed to save this as not trim.sh. I mean, you can save it as trim as your list.txt. So let's save it as list.txt. Control X.
Okay. Control L X.
X, yes. Yes. So delete this trimmed, yeah. So save it as list.txt. Do you understand? Do you now understand the way this list became about this list now?
Yes.
So for now, don't bother about the theory of the biology. No, you have to. I want you to get the technical details of the program itself, so that once you understand the program, you can tweak it to whatever way you want. So press enter. Yes. Now type ls. So you can now try to cut list.txt. Let's see what's inside list.txt. So now, when we finally create our trim.sh, it will be looking for this pattern and now combine this pattern or add this pattern to something underscore R1, whatever you have in the .fastq file. So that's what it will be looking for. And that's why this will be looking for, will be matching with, is it 335? No, it will be matching with this and then add the remaining part by itself, you see? But what you guys were doing before is that you guys will copy all of this and now tell it to be looking for this. It's really fun. You copy everything and still tell it to look for this and add this again. So you now have the full length plus the partial length, and that will be really confusing for the computer. So it will tell you no file found. You see? So now we've created our list.txt. Now let's now create our trimmed.sh or whatever you want to call it. Do you understand the process now?
Yes, I do.
Yeah. I would say advise that after now, when we are done, delete everything and do it again.
That's risky.
No.
Okay,
like after the whole tutorial today, delete everything and come back again.
Okay.
Yeah, it will just reinforce the learning. That's all. So go to the tutorial and copy that code, the entire code.
Okay. Oh.
No, not here. At the trimming stage. When you are tired, let me know.
No, I can't be tired.
So copy up to make directory, from make directory here, and stop at done. I don't think we need multi-QC so much. Yeah, we need it, but it's not a priority for now. You can come and do it.
You hope it won't affect this FASTQ. Okay.
So what do you think the multi-QC is doing?
It's to aggregate all our FASTQ
so,
reports.
yeah, but I'm not going to do FASTQ. That's just what I'm telling you.
Okay.
It's not. Yeah, it's good to check, but I know it's going to be correct. That's why I'm skipping it.
Okay.
So for reporting purpose, you should of course do it, but for the purpose of time and every other thing, please we can skip it.
Okay.
So you have 8 threads, and you have yeah. So hopefully it should work. If
So
it does not work,
okay.
Yeah, what you have a question? Say it.
Yes, since you said in our cat list we didn't specify this, so
Yeah.
but we are still using it. Is it? And we are saying that it should look for this. I mean, is that what we are saying? Okay.
Do you remember what I said?
Okay. Yes, I remember the sample.
Yeah.
Okay.
So it's going to look for that pattern and
Okay.
add it
And
to,
add this
yeah.
one. Okay.
So
I should
press save. Hopefully it works. If it does not work, then bash. So our advice, every time you get down, please shift it up. Always shift it up. Control L.
Okay.
Just press Control L. Yeah. So that we can see it. Not everybody has. Who is that?
Does not exist.
Yeah, what does not exist? You need to know what does not exist.
This.
Before you get there, there was there was error somewhere else. You should read your logs from the beginning.
Oh.
So yeah, prefix, then exception, file not. So this is how you read your logs.
Okay.
You have where did the command stop? Yeah, so this is where the command stopped. So it's identified the prefix of the Trimomatic, then luminal clip, yeah, but exception in thread, file not found exception. Hold on, I'm coming. I have to drag my the file not found. Then
I think it's the
did
raw
not find
data, but
raw data.
he's in raw data and he's linking back to raw data.
Exactly.
Yes.
So you have to delete the raw data in the. So quickly edit it. Yeah, just you can't use your mouse. Sorry, young man. Why do you want to keep the slash?
Okay.
Yeah.
That's. But it gave one error about ASCII
because it could not find the initial one. Others will fail.
Oh.
Actually, you have FASTQ. Yeah, you should actually. We don't really need the FASTQ sales, but you can go ahead with it if you want.
Okay.
You see, it is now working.
Oh, that is not the one that takes.
Actually, it takes time.
Oh my goodness.
It takes time, but we can stop it.
No.
I mean, we can still use the unprocessed data and go forward with BWA if you want, or we can stop with you and continue with another person that has done this step.
All right. That's all right, sir. But this is not where I had problem actually.
Yeah, I have to take you back to the. So you watch from the next person.
Okay.
So who's the next? I mean, who has done trimming already? So anybody that had issue with trimming can already study. I mean, I think Amara also had the same issue. So you should probably look at it and compare it with yours. Who has completed trimming so we can?
I have.
And but you have done your mapping already.
Yes.
So we can use it. We want to continue. So when we get to someone who has done trimming but has not done mapping.
Okay.
Yeah, after that person, we can use your own data. Who has done trimming but not mapping? I mean, who has stopped at trimming, or who is stuck on mapping right now? Okay, it seems mapping is very easy for everybody. Then so there are some files in the mapping state that I actually wanted to sorry. Yeah, maybe yeah, maybe Francois, we can use your data then.
Okay. I should stop sharing.
Yeah. We'll just go over the commands and see how we can make others like learn from the data. So Francois, if you can share your screen. Did somebody send me a message? Yep. And try to log in. Wow, the two of you are in Newton.
Yes.
One other thing, if you are, I think some people are done, some groups are done already, so I will just delete their folder. So
They've downloaded already.
Huh?
I said, oh, they've downloaded all their files.
I told them to download it. Okay, so Francois, let's start with yeah, the reads mapping. I think the reads mapping is actually very easy. Everybody can do it. Right? Yeah, you can show us the script. Cool. Cool. Can you just try to explain the script to us? I want to drink water now.
Yeah. So like here I created a directory named mapping. First, I indexed the reference genome that we downloaded previously.
So can you show it according to that you were according in a way that it shows that you are following the tutorial? Just
okay. Do I have to open this?
Yeah,
one?
show it on the tutorial and show it here basically.
Okay. Okay.
Yeah. And
so
if
like
there are any, sorry for cutting you again, if there are any things you edited just to fit your own kind of data, then you can show us again.
Okay.
Yeah.
So like for mapping, we created a directory named mapping. And then after that, we performed the alignment. So we did it using BWA. And here they added this, with the flag R, they added this this header here. And then they put the reference genome, the trim read. So remember in a previous step, we have to trim the read using Trimomatic, and the output that we have to use them for mapping.
Yeah.
And we will save our output under the directory we created named mapping.
Yeah, so just one thing, to put it into proper context, Temitope created a folder called trimmed reads. Sorry, I would annotate your screen. I love to. So Temitope created this folder at its Trimomatic stage and then sent the files into here. I mean, into Trimomatic. Into trimmed reads. So he sent the files. So the files that were created inside trimmed reads are these, basically. So what Francois is telling you is that you have to specify the folder you created in the previous stage, that is the stage Temitope is doing currently, and then push your files into the trim reads. And then when you are at this stage, you have to mention these files in the context of trim reads. That is, you have to first of all specify trim reads, then put the slash, then put it. Hopefully that's clear, if I did not confuse you. If anybody has questions, please ask. Yeah, you can go ahead now, sorry.
Okay, so we save our output under the mapping folder. This one is for the normal sample, and the second one is for the thermal sample. So this is basically how we do it. So like, do I have to run it again?
No, don't worry.
Okay.
You already have it.
So this is the script that I used to do it.
So basically, it just worked.
Okay, yeah.
Yeah, that's cool.
Can I ask why this flag? Why this backslash?
This one?
This yes,
one?
yes.
Because we are running like, I think this is just to specify that we are using different lines, if I'm not wrong. Yeah.
It's supposed to tell you that it's still the same line. You are like, I mean, yeah, you are splitting your lines into two, but the code should consider them as one. If you want, you can delete this space, you can delete everything and keep everything as a single line.
Yeah.
It's still the same thing. But so that you can, for readability, yeah, that's what people say. So that you can read it, and the command does not consider it as a next line, so to speak. So you can put it there. Okay, thank you. Next, so what was the next thing you did?
We have to convert the sum file to BAM. Sort them and index them. So this is what we use. We will use the list.txt file created before. And then first we have to like read the file, I guess. And then after, we have to convert it into a BAM. The flag B here is used to convert the sum file into BAM.
Perfect.
And then we have to sort the file and save the output under mapping with this extension, sorted.
Yeah.
And after that, we have to index the file and save it under this. We have just to index the sorted BAM file.
Yeah. So before you go ahead, so I just want people to note that this mapping, this folder called mapping, was created in the previous stage. Right? Am I right?
Like, yeah.
Yeah, was created here. So so I'm trying to let you know that you can actually change that mapping if you want. You can change it to Joseph, or you can change it to whatever you want, or you can change it to your name. But when you are mentioning it here, you have to mention that thing you use, maybe Joseph or your name. Okay. So it's a sequential order. You can do this thing completely different from how they did it here, using your own kind of variables. You just have to follow that. Okay, when you are specifying this file, you have to specify, when you are specifying this file, you have to remember it for the next step. When you are specifying this folder, you have to remember it for the next step. It's basically coming back to recalling what you did already. So yeah, Francois, you can go ahead.
Okay. So like after this one, after indexing the file, we have to filter the map read.
First of all, did you change anything here at all at this stage?
For here?
No, just separate something. Did you change anything at all?
Let me see.
Yeah. Yeah, you have to exit here. Control X.
Don't know why, but it's not working.
I guess you are logged out already.
Server is closed, yeah. Okay. So this is basically what I did. So like to sort, you told me that I have to sort by position, because if we don't do that, when we are removing the duplicate, we will have an error.
Yeah.
So I sort it by names
Yeah.
instead of position. Yeah. This is the only change that
So
I did.
did you export all your codes out already?
Not yet.
I think you should, and save it for your personal use.
Oh, on my local computer, yes, I did. I have that here.
Okay.
Yeah,
That's cool. So you can save it and you can put it on GitHub. You should,
okay.
You can. You should put it on GitHub. And your GitHub profile is one of the things we would look at the end. So all these codes, you can put them there. And you can share it with others if they if they need help from you, you know. Or you can, anybody could request for help. And you can even recreate this tutorial to your own taste. Yeah.
Okay.
Yeah. But these things are things you can do later on. Okay. So
after indexing the...
Sorry, I have a question.
Okay.
Okay.
The line, I'm seeing flag N. Why?
Yes. The flag N just means that we are sorting sorting the file by names.
Okay.
So would it affect my output if I don't include it?
Well, I tried sorting by position, but when I run the remove duplicate, it won't work. So they say that I have first to sort them by position and then sort them when removing the direct duplicate, sort them by here I have, sorry, to sort them first by name, and then when removing duplicates, I will sort them by position.
Yeah. So let's go back to the tutorial. Let's check something. So I think there is an update in SAM tools, and we probably installed the latest SAM tools. This tutorial was done, I think, last year or 2 years ago. I can't remember. So that's one other thing you need to know about bioinformatics software. They update them, then they are being updated. And the fact that you know it now does not mean you use the same set of commands next year. So I think you can, before now, you can sort without the flag N thing, but now I think you just have to include flag N. So one thing you can do is to probably send a mail to the authors of this thing and tell them to update their tutorial. It's not from ArcBio, but they were fellows like you guys last year. You can always write to people, you know, but you just have to be polite. You don't have to say, hey, you wrote a stupid code, and you can just tell them, I noticed that there is an update in some tools, and I will be really happy if you can fix some of your codes. They were having some bugs already because it's no longer compatible with the current edition of some tools. You see, that's more friendly, and you can speak to the person that way. So yeah, the flagging thing is very important, and I think you have to put it back to default later on, right? Am I right, Francois?
Yes.
Yeah. Okay.
Do I have to?
Yeah, go ahead. Yeah, go
okay.
ahead.
So like for here, we are going to filter the mapped reads, the one that we have from the alignment, using some tools view. And this one, the flag queue mean that we will consider only reads that have a base quality score greater or equal than 1, if I'm not wrong.
Yeah.
And this one, we will consider only the reads that are mapped to the reference genome. And we will remove the reads that the reads for which the mate are not mapped to the reference genome. We won't consider them here. We will filter them. And then we will save our output to a sorted BUM file, a filtered BUM file. the sorted BAM file will be our input file.
Yeah. So why were you having issues? Was it this one that you had issues with?
Me?
Yeah.
Okay, it actually worked for me. So I have an issue during variant annotation with Gemini.
Ah.
Yeah.
Okay, but
So I don't know. Maybe I have to like check the files.
So we were trying to fix something around duplication of. So was
it was the size of the file. I had like 92 bytes for the size. So you told me that I have like, I was doing the job twice since I had like the sample's name written here twice. So I just did it like you told, and it worked.
Sorry. I'm trying to look at our charts.
So,
I mean, our charts where we.
I think.
Ah, okay, I see. I think it was around. So you had 4 things in your sample. I mean, when you were trying to create how will I call it? When you were trying to create the samples.
Yes, when I was trying to sort the file, so
okay.
This was where I was having problem.
I see.
It said that coordinate sorted, required group, and sorted by carry name. So
okay. Then let's go back.
Okay.
Sorry for the constant break in your transmission.
It's okay. So like for this step, they said that we can see the results of our alignment here using some tool Flagstaff, but I didn't use it.
Hold on. Yeah, some of these things are just they're just fulfilling your righteousness. Actually, you really don't need to be too stringent on the data set like this. Yeah.
Okay. So the next step is here, removing the duplicate. So like here, I was also having a problem with the dash O. When using the dash O, it gives me, it gives an error saying that I can't use the flag O. So basically, let me just see the file. So here I didn't use the flag O. So I just first put my input BAM file and then give the output BAM file here.
Yeah.
And then after that.
Yeah, so one thing, this was a major problem. I think we fixed it together with Judith yesterday as well.
Okay.
So we had SAM2's collate flag O previously, but this flag O, you guys need to remove it. So I think what you guys can do really, maybe we can team up some of you, I mean, some people that have solved this step, is to create a list of the codes that are working. And you guys can now have your own repository and publish it as a correction to, to the old one, you see? Yeah, so I think everybody should take note of this. So I will show you something. No, I can't show you, don't worry. But I mean, I wanted to show you the SAM2's repository because initially there was flag O, but I think there was an update. So they wrote that the flag O, this thing has been deprecated and you don't really need to use it again. So now SAM2's collate works with first the inputs, then the name of whatever you want to create at the end. I think, yeah. Am I right?
Yeah.
Yeah, okay. So you have to have this filtered one first, then name collate, and then samples, fixed minutes, whatever. Okay. So it's a very important step because this was where most people were getting errors. Yeah. Yeah, go ahead.
Okay.
Yeah, sorry. I had an error in this. Whenever I tried to do this particular command, the samples collate command, I used to initially get the error that the small o should not be used and capital O should be used.
Yeah.
I tried that, but it did not work. And right now I saw that I should remove the flag O. I just now I tried to remove the flag O, and then I tried to run the command again, and it is saying that failed to open file mapping slash name collate dot bam. So
do you have name collate first or filtered one first?
I have the filtered one as the file that is there in that. Sorry, in the command I have name collate first and then filtered as the second.
Swap it.
So I have to interchange.
Yeah.
Okay.
Yeah, you just have to swap it because filtered one is the input and the name collate is the output.
Okay, let me try.
Let's know how it goes.
Okay, so after that we will remove the duplicate with the dash R here.
Yeah, so I'm playing with your screen.
Okay. Okay.
This thing is not stopping. Yeah. So this is a very important thing, and it's not your fault. It's not anybody's fault. It's just that there was an upgrade between last year and this year in some tools, and it worked for them last year, but it was no longer working this year. You see? Yeah. So it's just we are learning, right? Yeah. So next, Francois.
Okay. So like here we have to left align the BAM file using BAM left align. So we will read the content of the clean BAM file and then pipe it to BAM left align. Here the dash F for our reference genome. So we are aligning against the reference genome. And then we will save the output under mapping left align that BAM. Okay, so here they said that the flag C mean compress and the flag M the max iteration. I don't really know what they
yeah,
mean.
maybe the number of times you have to.
Yeah,
it's like iteration is repeating the same thing. It's like a loop.
Okay.
Yeah, some people call it loops. So it's like repeating or going over it again. So some of these things, there are lots of computations that go inside. For now, just be bothered about the accuracy of the codes. I mean, yeah, you can be interested in the technical details of the coding, but you should be fine.
Okay. So after left align the files, we have to calibrate them using some tools, count the input BAM file, the one that we have after left aligning, and then the reference genome here, and we will save our output under mapping, adding the extension recalibrate that BAM. So here also I did not change something here.
Okay, you did not change anything. That's cool.
Yeah.
Yeah, it's good to note that there are points where we have to change a few things, and there are points where you just have to follow.
Yeah.
Okay.
So after that, they filtered the read mapping qualities. So using BAM tool filter, and then they save that under mapping prefilter that BAM. So here we have to add codes.
Yeah.
Because if we do not, it will consider this as a file or a directory. So
I think this is one of the new things that changed in SAMTools again.
Yeah.
Yeah, usually very tiny things, and if you're not careful, it just mess you up and takes your entire day. And that's why it's good to ask questions. Okay. It's a program updates, software updates is a general problem in programming generally. So there's nothing we can do about it. You just have to keep yourself up to date. That's all.
So here I didn't change it, so I did the changes here. So like basically you have to like add the codes here. I have to change this. So after that, we have to do variant calling using varscan somatic. So we will first download the code using this link wget. I think that
so about that, varscan has been installed already.
Okay.
Yeah, so you don't really need it.
So okay. For me, I just downloaded it on.
Yeah, if you have to download it every time, everybody will be downloading them.
Okay.
Yeah.
So we have to convert the BAM file to pilot using some tools and pilot, because the pilot files should be the input for varscan. So we have to convert them to pilot using some tools pilot, the reference genome also, and then our input read, the filter read BAM file, and then we will save it under variant. So we'll have to make a directory variant and then save the output to variant with the extension .pilot.
Edit anything in the codes as it is?
No, I don't think so.
Okay.
I didn't.
Okay.
So this is it. I didn't change anything here.
Okay.
Okay. So after that, we have to like call the variants with varscan. So this is the code. I don't know if there is a change since I downloaded the files under.
So you can actually redo the data. I mean, this command, you can actually remove this.
This thing?
Yeah, you can remove all of these.
Okay.
So varscan alone will work. Let's go to the terminal. Let's see if varscan will work. How long did it take you to complete it? This varscan stage, how long?
The time?
Yeah.
I don't really remember, but
it was really long.
I think so. I just use my screen and run it and like start doing other stuff.
Okay, but so what I wanted to do is that so type varscan. I just want to show others how the command will look like, so that when they are watching this record later, they can just clear.
Okay.
Yeah, so varscan, capital V, I guess, I'm not sure. Tab. Use tab to fix there.
Okay.
Did it work?
Let me see. No.
Okay. Press N. AR. VAR.
VAR. Yeah.
Yeah. Tab.
It didn't work.
Yeah. So change it to capital V.
Okay. Okay. Now it works. But this is the folder that I downloaded, I think.
I see. Hold on. Let me check on my end too. I know we have it, and some people used it actually. Oh, sorry about that. So, yeah, the small letter works for me. var scan.
This one, it's this one.
Yeah, it works.
Yeah.
So yeah, type it again. Var, no. Var, yeah.
Can I use the help to see if it's...
Yeah.
Okay.
Maybe just flag H. Yeah, okay, it works.
Okay.
It's there.
Yeah.
So yeah, you can now go back to your terminal. I mean, your tutorial.
Yeah.
So instead of writing the Java, whatever, just ignore this and ignore this. And this should now be small letter VAR.
Okay.
And I can just type it. VAR, SCAN. You see?
Yes.
Var scan. So it's so you should just replace this.
Okay.
Replace this with with this guy here. Yeah. That's very good. You can actually, this is very good. Yeah. So I could not identify the person that mentioned the flag O issue. Has it been solved? I didn't see the name of the person. Yeah, so you need to remove this dots to whatever. Yeah, this version and jar. And the scan should be small s. The var scan. Yeah.
Oh yeah. Okay. So it's okay now.
Yeah.
Okay. So this is the step where we will merge the file generated with varscan.
Yeah.
So these are the file snp.vcf. We will save it under one single VCF file. So it will be saved under variants, the folder, the directory called variants. And after that, we will start the annotation using SNPF.
So SNPF also has been installed.
Yes.
Yeah, so you probably don't need the Java thing. So Francois, I don't know, but do you need the mapping files anymore? My advice is that you just delete all of them to reduce space they are using. I mean, I think you should look at, oh, he deleted them. Some of them must have gone. I wanted you to look at it and see if there is something you need.
I already downloaded the.
Then just wipe it out. Once you download things like that, please wipe them out. Okay. Yeah, then this fast QC report as well, they can go. Yeah, I removed trim grids as well. Even the raw data, we really don't need them. They are just big files. And I think every other person can do it. Or else I'll come into your folder and delete it myself.
So do I have to delete this one also?
Since you unzipped it already, right? And we actually have it on the folder, so I mean on the server. Latest. See, hopefully we have more storage space now. Yeah, I think you're typing something different from the var scan. You only wrote var can. Yeah. Okay. Yeah, I think we have more space now. Thanks to Francois for giving us more space on the server. Then
this the functional annotation with SNPF.
Yeah.
So we have to download the file. So since it's already available, we won't. And I don't know if you have to download like the database.
Yeah, I mean, you can download it.
Okay.
Yeah, you have to download it because I don't know how to create a file that everybody will be using. Sometimes there will be permission issues. Yeah, downloads. Yeah, not everything works that way. HG19. Yeah, so you are downloading it. Oh, you are trying to recreate the tutorial?
I already did this. So
yeah, so you have to stop it. Stop it, don't worry. Yeah, we don't need another copy of it. So basically what it's telling you is that it's going to work.
Okay, so this is the step where we have to annotate the variants.
Yeah,
so
this is the most important one.
We will use, I think, this command instead of adding this.
You have to, exactly, and you have to delete, you don't need this initial part. You don't need
this one. Oh, yeah.
You only need
SNPF. Yeah. So I don't know if I have to run it since I already.
No need. You already did it.
Okay. So after, when we run this, we will save it under variants and with this name.
Annotated, yeah, ANM.
And perform the clinical annotation. So this is the step where
So you are not doing this step on the server, like we said already?
Yeah, I tried to download it on my computer, but it's giving me an error.
You tried you cannot download it?
No. I downloaded the installation file, the Python installation file. But when I execute it, yeah, it gives me an error. It won't run.
Yeah. So what most people did was to do it on the server. I mean, on the Galaxy server.
Yes.
Yeah, that's what most people did. And I would advise that you try the Galaxy first. But really, for me, if you are able to produce your VCF files alone, you are more than enough. I mean, yeah.
Okay, but using also the Galaxy server, I had an issue there. So
okay.
When doing the annotation, like we have to use different steps to annotate our files. But when I try to do it, there is a step where I have an error.
Yes. Actually, I'm not a big fan of Galaxy myself. I don't use it. But there are some people who love it. Yeah, but I can't see errors here.
Well, I deleted the file with.
Okay.
So we were asked to, let me just open the link to the tutorial. So here we were asked to download these bed files. And when doing the annotation, they give us a file that do not exist in our data sets. So just go there.
Which file?
1 second, please. This one, the cancer hotspot underscore v2.bed. We do not have it on our data sets. We only have hotspot that bed file. So if I try to use that file, I have an error. And if I skip this step also and came to this one, I have an error also.
Hi, can I help?
Yeah.
Okay, can you go back to the Galaxy work?
Yes.
Please? The step for the cancer hotspot.
Okay, yeah.
Can you repeat the particular step? There's a part where I got that same error too, and there's something I did.
So I have to repeat all of them? Or
No, just the hotspot. You still have those data sets, the bed files,
yes,
right?
Yes, I do.
Yeah, just repeat that. Okay.
So this is the database that I use. So here I have to select the hotspot. This one. Okay. So let me see what I have to add here.
Is it just me? Just okay. Let's say your screen is normal.
So here I will add 5, right?
Check. I don't have it offhand. Just check the tutorial.
Okay.
And is everybody using this Australian Galaxy?
No, mine wasn't Australian. Mine ended with EU.
EU, yeah. Sometimes the things found in it in different Galaxy versions are different. For example, I was also working on one African version once upon a time, but there was no support and the whole thing just died.
Okay, can you scroll to where your file is, like the hotspots file? Just let it continue running. On the right, your right, yes.
This one.
Yeah.
Okay.
Click on it.
Do I have to view it?
Edit.
Oh, okay. Edit. Yes.
Okay. Database build, unspecified, down.
Okay, this one, yeah.
Yeah. Click on it.
I think almost
It should be human there.
it's just type HG19, it should, yeah.
Okay.
Yeah.
This one.
Yes.
Okay.
That was the problem. So if this still flags error, so you can rerun it with the updated files. It should work.
Okay, thank you.
You see? You see why you should ask questions, guys?
Yeah.
So when you ask questions, maybe I don't even know it because I'm not a big fan of Galaxy myself. It's a good tool. It makes computing available for everybody, but I'm not really using it. See, like I'm not always using it. I have Linux servers I can always use. So I would rather just do it on my Linux server and forget about it. Than, yeah. So yeah, different people have different experiences. So I would advise that apart from your teams, you can also drop these comments on the general chats, and people will be willing to help you. Somebody like Halimat can help you.
Okay, so I think this is all for me. I have to wait for.
Yeah, you have to wait now.
Yeah.
Did anybody have any other problem after here?
Hello, I also had the same issue that Francois had with the Galaxy.
Ah, so you see your solution now?
Yes, so I'm also trying it.
Yeah, that's
good. I wanted to ask that are we supposed to change like all the bed files?
Change?
The data types, should we change everything to human? All the bed files?
Yeah, maybe you should.
No, not really.
Okay.
No, it was just this one that had that issue. I think there was a part in the error where they actually mentioned what the problem was. So I had to go to the file attributes to check.
You see? Are you satisfied now, Eniela? I think.
Yes, I'm satisfied. I've been stuck on this for some time.
Sorry.
Sorry.
I think you've been trying to chat me for some time. I've not
yes, and you've not been answering me.
So last week was just something else. Don't worry, this week I'm much more available and we would work on the project together, because this is going to be one of the first things that is going out of ArcBio. I mean, through you guys. So it has to be perfect. So I will be on your necks.
Stop the calls.
Yeah?
I would stop you with calls.
You will disturb?
I will disturb you with calls.
Ah, yeah. Maybe I will not allow you. You can always use chat first. Let's try chat. Then if I see that the chat is not working, I will suggest a call. Because I'm probably responding to not just you. So, I mean, maybe you can write to Michael sometimes as well.
Yes, I write to Michael.
Yeah, you should be able to help.
He said he's not really familiar with this Galaxy.
Yeah, the thing is, the Galaxy thing is not really so. There is something generally. It's one of the problems of programming and data. I mean, anything technology. One of the problems is that people immediately put ranks amongst themselves. So if you are a developer and you are a developer that does not write codes, maybe you are just using already used softwares to build your websites, people will consider you second class. They will not want to say, they won't put it to your face, but they won't value your hard work. They would say, oh, so you're just dragging and dropping. But why they are doing the real hard work of coding it out. Yes, Galaxy is like Canva, and the Linux thing is like maybe Photoshop. You see, it's like putting yourself in class. Like, so these kind of things exist, and to avoid that kind of discrimination, it's good to learn the Linux. And maybe when you are short of resources, you can use Galaxy. Okay, I think so far nobody is.
I have an
Okay.
Yes, so I tried querying my data, but it's not coming up. It's not bringing out anything. It's just creating an empty table.
Yeah, somebody faced this.
Yeah. So I reached out to the person, but the person said they started all over again using Galaxy.
That's not good. Can you try this thing that Alimat suggested now? Or you have tried it already?
Yeah, I've annotated, I've passed all those stages. It's just, and I'm getting, those stages are going through. But like doing the quick. Yes.
Okay. So.
Yeah, so this is, so I've loaded it. So this was the first query I did on the data set, but it's empty.
Can I see the step, I mean, where you did it? I mean, the things you put in here.
Okay, so the first one I did, so I had to do this first on my data set. So I loaded this data set into Galaxy because I used the Linux pipeline. So after loading this, I performed this on it. And this was what I got for this data set.
Yeah, let's see this one. Let's see. Okay, there's something there.
Yes. So after performing this, I went back to load my data set.
And it was successful?
Yes, but I can't open this. It keeps saying I should download, so I'm not sure.
Yeah, it's SQLite data.
Yeah. So it looks, it's 219 MB.
Yeah. Next.
Then I was supposed to do, annotate the loaded, the same thing. So it's updated, it has variance data in it. Then the last one.
So the last one is what is giving you problem now? Like,
yeah, so the last one, no, I'm able to do the last one. What is giving me a problem now is the query, where I'm supposed to produce a table.
Yeah, so let me see where did you do the query thing? Like, what are the inputs for the query?
So the input for the query was my last annotation.
Okay, show us on, like, demo the way you do it.
Okay, so let me go back.
probably still the same problem. Although my own data set started with like 10 lines. This one is only having 1 line.
You said what?
I said it's probably still the same problem because.
Okay.
So it says, so starting from the loading. Sorry. Yeah. So it says I should load this, the output of this, which I did initially. So my output, yeah, 32.
So, yep.
And annotate with this. Then.
So hold on.
Yeah.
Before you come here, yeah, go back to Galaxy. The first file you used, let's scroll up. Yeah. So locate it on, it's number 31, right?
Yes.
So edit it using the edit icon. Let's see the attributes. Yeah, so change database and build.
I changed the database to
HD19. The same thing we did just now. Yeah. 2009. I mean, there are upgrades, but yeah, that's enough.
Yeah. Which one? Which of them?
2009 February. Yeah.
Okay.
No. This is upload type. So, so I can also talk about the human chromosome build and the reference chromosome. Sorry, let me.
Is it the homospecian?
No. This human February 2009. You already created.
Yeah.
Yeah.
Absolutely.
Yeah. Then save.
Okay. So can I go ahead and do the query, or do I need to?
Yeah, let's see other files that you used. We'll do everything. We'll do for everything and see what happens.
Okay, so
go back to all the files that you use for that step. Let's try to fix them. HG19. Yeah, you've corrected this.
Okay.
So the database is already HT19. See the cancer hotspots too. Or you don't need it actually.
No, I'm still okay.
Yeah, you finish this one. Finish.
Yes. Yeah. Yes.
Okay, now let's try it again. How long does it take on average?
Today it has been really slow. The server has been really slow, so
it's because of you people.
Well, no, it doesn't take so long, like maybe 5-10 minutes. But today I can't guarantee.
Okay. Let's see. If you want, if it's taking too long, then you can just share the updates about it on the team.
So I don't need to do the steps again, or can I go to the query?
No, just go straight to the final query you said you giving issues. And if it still gives issues, then you can try to start from scratch again.
Okay.
So who has started the general, I mean, you can stop presenting. I think you know what to do.
Yeah. Thank you.
Projects, you're welcome. Has anybody started a project? Okay, you guys are still on the stage 2. Stage 2 is taking 2 weeks. It's living up to its name. That's good. Hopefully you guys can complete it by the end of today, say tomorrow max, and then you begin your projects in full scale. Okay. Any other question? We spent 3 hours, 3 good hours. I mean, is it 3? No, 2 hours. I started 4 and we are closing by 6. That's good. So you stayed up to time. And so no questions.
I have a question.
Yeah.
So for the reports we are supposed to submit, are we submitting it as a group or as individuals?
As individuals, then you collate the list of links to the PDF. Maybe you upload it on your GitHub or something. I can show you the way others did it.
Yes.
This is from the other team. Sorry. I think it's as a group, I think, and you list the contributors here. So this is what I did. You list the contributors. I'm not supposed to share it with you. But it's okay. So you do it as a group. But if you have your own reports ready or you want to write it on your own, let me know. But I would advise you work as a team. Yeah, it's better you work as a team. It's a long report. And everybody can review, you share ideas on the meaning of what you actually did. Any other question? I mean, if you still want to ask a question, you can stay, but the call is officially over and I'll stop recording now. Bye.